For this chapter, we're talking about chemotherapy, trying to kill microbes inside of the human body, which is significantly different than what we talked about before with chemicals and physical agents to try and kill microbes outside, in the outside environment or on the surface of the body. In this case, you want to give a drug that can kill off the microbes without hurting your patient. Now, the birth of this whole idea, modern chemotherapy, came from a guy named Alexander Fleming. Alexander Fleming was a British doctor during World War I, and he was horrified by how many soldiers died of infection. In fact, more soldiers died of infection during World War I than died from battle wounds. And he was just absolutely horrified by that, and he wanted to find a way to kill specifically the microbial cells that were causing the infection without hurting the patient. So he was looking for some substance that he could use, that he could give to the patients. Patients would swallow the pill and it would kill off the microbial cells, the infectious cells. Well, Alexander Fleming, after the war, went back to his research, and he was looking for this, and he searched and searched for years for some substance. Now, in addition, we're also talking about a time frame where people didn't have the little plastic petri dishes that we have now. The petri dishes were all glass, and so after an experiment was over, you would have to scoop out all the stuff from the inside of the dish and wash the dishes and sterilize the dishes and put in the fresh media. And sort of lucky for us, Alexander Fleming was not a very tidy person. He wasn't a very tidy lab worker, and he would allow these old dishes to just pile up. And eventually, when they broke down and washed them one time, he realized something very interesting, and this is really the scientist in him. Not only was he just cleaning dishes, but he was looking at this really strange phenomenon. And what he noticed is the bacteria he had been working on here, a little bit of mold had fallen onto the plate. Not surprising, mold contaminates plates all the time. But what he noticed that was so revolutionary is that something that the mold was creating was killing the bacteria around it. See that? There's this ring of area of bacterial death where the bacteria can't grow because something being made by that mold is killing off the bacteria. It took a while to find what that protein actually was that was being produced by the mold. Since the mold was called penicillium, he named that first antibiotic penicillin. It took a while for them to actually produce this antibiotic penicillin in mass quantities. By the 40s, it had been tested and was being produced and used to treat patients. And this was the beginning of the idea of chemotherapy, giving drugs to a patient that could specifically kill off the infectious agent without hurting the human cells. Now, his work really alludes to this idea, but the origins of antimicrobial drugs, almost all of the antibiotics come from soil bacteria or fungi. They're being made either by bacteria that are in the genera Streptomyces or Bacillus, or molds in the genera Penicillium or Cephalosporium. And what's happening here is different types of bacteria and different types of fungus are living in the soil, and they are competing with one another. They're competing for resources, just like any organisms who are growing in the same environment are competing. If we think, for example, about two lions, right? Two lions are going to be fighting over all sorts of things. They'll be fighting over food sources. They'll be fighting over water sources. They'll be fighting over access to a mate. And they will be doing things to try and kill off their competition. Same exact thing happens on a microbial scale. The bacteria and the molds living in the soil are making things to kill off their competition. And mostly these are soil bacteria and soil mold. So Streptomyces and Bacillus are the bacterial genera, and Penicillium and Cephalosporium are the mold genera that do this. And again, they're making stuff to try and kill off the competition and help them to survive and get more of the nutrients. So most of the antimicrobial drugs that we have actually come from some bacteria or fungus who makes it naturally as part of their attempt to kill off the competition. That's what we saw on Fleming's plate a little bit ago, the mold creating something to kill off the competing bacteria. In fact, to this day, when they search for new antibiotics, what they'll often do is take a very resistant strain of some sort of bacteria, make a lawn with that resistant strain, and then they'll sprinkle a little bit of a soil sample on top. And what they look for is anything where it shows that the resistant bacteria is being killed by some organism in the soil. So in the background here you can see the fuzziness, that's the lawn of bacteria. This organism is making something that's killing off the bacteria around it. This black organism here is making something that's killing off the bacteria around it. This great big one is making something that's killing off the bacteria around it. So what a researcher would do is take a sample from each of those and start to try and investigate what is it that's being made. And is it possible to use this to kill off bacteria or mold during an infection? So if we were going to come up with the perfect antimicrobial drug, what sorts of things would we need to consider to create this perfect antimicrobial drug? First thing, of course, the drug would have to exhibit what we call selective toxicity. Selective toxicity means that it will kill or inhibit a microbial cell without damaging the host tissue. All drugs have to exhibit selective toxicity. They have to be killing off only the microbial cell without hurting the human cells. That's absolutely essential, of course, for any drug. Also, if it was a perfect drug, you would want it to have long-lasting potency. It would be nice if you didn't have to have regular delivery of the drug, like if you only took it once a day instead of 4 times a day, that would be nice. If it could survive long-term in the human body. Easily delivered to the appropriate site of infection. We like to apply the drug to particularly where the infection is instead of having it circulate through the entire body. Doesn't disrupt the host's health. So it doesn't harm the host's health in some way. Now sometimes people think, wait, isn't that selective toxicity? No, selective toxicity is basically recognizing the difference between a microbial cell versus a human cell. With disrupting the host's health, I'm talking about things like allergies or other various complications that can occur, and you want a drug that has as few of those as possible. The last one here, doesn't encourage the development of antimicrobial resistance. Without a doubt, you guys are aware that we're having a problem right now with antibiotic resistances. And some drugs actually discourage the creation of antibiotic resistance. And so that's what we would hope for. We would hope that antibiotic resistance is not going to happen with our perfect antimicrobial drug. Now, of course, this drug does not exist. There is no such thing as a perfect drug. It's always a balancing of pros and cons whenever they come up with a new drug. These are always the things that are considered, but when it comes right down to it, nothing's perfect. We're always balancing these things out. So selective toxicity is an absolute requirement. After that, everything is a balancing act to try and make the best drug possible. All right, we're going to go through several different groups of antimicrobial drugs. I have color-coded this. Of course, microbes are very different one from another, and therefore a drug that kills one kind of microbe isn't going to kill all of the microbes, right? A drug that's meant to kill bacteria is not going to hurt a fungus, and a drug that's meant to kill a fungus isn't going to hurt a virus. So they are very different drugs for different kinds of microbes. So I have color-coded this lecture, so as you're going through, all the antibacterial drugs are in that purple color, the antifungal drugs in orange, the antiparasitic drugs in green, the antiviral drugs in red. Try and help you separate them. Now the antibacterial drugs we're going to talk about first. There are really 2 different types of antibacterial drugs, the antibiotics and the synthetic drugs. The antibiotics are drugs that are produced naturally by some microbe. This is what we were talking about a few moments ago. The ones that are naturally made by soil bacteria, we isolate them and use them to treat patients. the vast majority of antibacterial drugs are antibiotics, where they are normally made by a soil microbe. There are a couple, however, that have been designed and synthesized in a lab. Those are synthetic drugs. They're not made naturally by any organism. They were purposefully designed as an antibacterial drug. All right, if we look through bacterial drugs first, if we think about targets for an antibacterial drug, there are several. And you guys actually had an essay question on the first exam that was about these sorts of targets and what sorts of things make good targets in an antibacterial drug. And of course, cell wall synthesis, i.e. peptidoglycan, makes a great target. Inhibiting nucleic acid synthesis, some of the enzymes that are used in DNA replication are different. So if you can target the bacterial version of that enzyme without harming the human version of that enzyme, you can inhibit the nucleic acid synthesis or replication. Inhibiting protein synthesis, there's a bunch of drugs that will target the 70S ribosome, some component of it, and that will inhibit protein synthesis. Or disrupting the cell membrane can also harm that cell if you can kill off that cell membrane. Now the first of the drugs, the antibacterial drugs, I'm going to present the first one to you. The rest of them you guys are going to present to each other next week in your peer teaching activity. So the first one here, antibacterial drug, penicillin. Penicillin, of course, is the first one that was identified by Fleming. It is the original for all of the antibiotics here. The penicillin, the drug penicillin, what it does is break down peptidoglycan. Let me show you how. Remember the structure of peptidoglycan here. We have alternating strands of NAM, NAG, NAM, NAG, NAM. We have multiple strands of NAGs and NAMs, and they are connected by little protein crosslinks. They're showing that in green in this diagram. What penicillin does is gets rid of those protein crosslinks. So down here, after you've been treated with penicillin, the bacteria no longer has those little crosslinks. That breaks the whole peptidoglycan apart. So it blocks cross-linking of the NAG-NAM strands. So if you look at it from the cell's perspective here, with normal peptidoglycan, the cell is strong and healthy. Exposure to penicillin causes the cell wall to break apart and the cell bursts and dies. That's how penicillin kills. Here you can see normal staphylococcus. If the staphylococcus is treated with penicillin, the cells burst because they don't have a cell wall any longer to protect them. For this reason, in general, penicillin is most effective on younger cells, and that's because younger cells are when they're developing their peptidoglycan. So penicillin tends to work best on younger cells as they're actually forming their cell wall. Now penicillin, the original penicillin that was discovered by Fleming, only works on gram-positive bacteria. It couldn't get through the outer membrane of gram-negative. So that original penicillin is specific only to gram-positives. They have developed newer versions. They call them the penicillins. This is a group of drugs that are based on the same structure as penicillin. They all end in -cillin, like amoxicillin, methicillin. Those are all in that penicillin group where they have a similar structure to penicillin. The nice thing is the rest of them in this group, the penicillins group, they have a broader spectrum. They can get both gram-positives and gram-negatives. Let me summarize that. The original penicillin was only gram-positive. The group penicillins are broad spectrum. They get both gram-positives and gram-negatives. Now, the penicillins are non-toxic. They do exhibit selective toxicity because it's targeting peptidoglycan, but unfortunately they can trigger allergies. There are a group of people who are allergic to penicillin and all of the penicillins. If you are allergic to the original, you shouldn't take any of the penicillin group. And in fact, if you're allergic to that, you would also be allergic to the penicillin mold. And by the way, it's the same mold that's used to make blue cheese, so you probably don't enjoy blue cheese dressing if you are allergic to penicillin. All of those go hand in hand. Or if you thought you liked blue cheese dressing and you found that you always got sick when you ate it, now you know why. If you're allergic to penicillin, you shouldn't be eating blue cheese. Now, unfortunately, some bacteria have evolved an ability to be resistant to penicillin. Here's the structure for penicillin, this antibiotic. Some bacteria have evolved an enzyme called penicillinase. Penicillinase breaks down penicillin and makes it inactive. In other words, those bacteria that have the enzyme penicillinase are resistant to penicillin. And so we're beginning to see lots and lots of penicillin resistance because, of course, once one bacteria has that gene, they can pass it on to others. Now, some penicillins aren't sensitive to penicillinase, so they can be used to treat penicillin-resistant infections. So, oops, I'm sorry. So, I'm trying to move this out of your way. There we go. So, for example, they originally developed methicillin in order to treat penicillin-resistant Staphylococcus aureus, but unfortunately now it has become resistant to methicillin as well, unfortunately. So that's another case where the more we use drugs, the more they become resistant. I'll talk more about that in a little bit. So the take-home message for penicillin here is how they kill by interrupting peptidoglycan. What they kill, the original penicillin was only gram-positives, but newer ones are gram-positives and gram-negatives. Unfortunately, they can cause allergies, and even more unfortunate, some bacteria have evolved an enzyme to get rid of it. So therefore, they can break down the penicillin. Now, this is the one drug that I'm going to present to you, and I gave you much more detail because it is a big group with lots of antibiotics in it. But you don't need that much detail about all of the different drugs. Remember, you are responsible for that peer teaching activity. You're responsible for presenting 4 drugs. I assigned those drugs to you last week in lab. So as you look at that list of 4 drugs, you're supposed to be telling us, 1, what it kills. Is it only gram-positives? Is it only gram-negatives? Is it only fungus? What does it kill? And how it kills, or how it exhibits selective toxicity, how it's destroying just the pathogen without hurting the human. If you notice something interesting about the drug as you're doing your research, by all means, bring that information in to the other students. It's always easier to remember something if you find something interesting to tell them. All of the information you should need is in your textbook. You shouldn't need to do extensive research outside of your textbook to find this information. So I'm not going to go through all of these other drugs because, again, we're going to do that in our class activity. I would suggest, however, before you come in for the class activity, what you should do is print out the lecture notes for this lecture so that you can fill in with the information as you learn it from the other students. So I would strongly suggest bringing in those lecture notes so that you can go through and fill in for each one of these drugs what it's killing and how it's killing. Now I'm going to skip through those drugs to the next topic here. Saving the drugs for next week, but I do want to talk about the human side effects of drugs. Now, of course, the goal with any of these drugs is to exhibit selective toxicity. We want drugs that are not going to hurt the patient, but are going to hurt the bacteria. Unfortunately, even though it may offer selective toxicity, sometimes there's cross-reaction. Some drugs do give some toxicity to the patient. For example, chloramphenicol. Chloramphenicol is a very effective killer, but unfortunately it also can hurt human bone marrow. Bone marrow is responsible for producing blood cells, both white blood cells and red blood cells. So if you destroy the bone marrow, you wind up with a form of leukemia called aplastic anemia. So chloramphenicol, unfortunately, its use has been recognized to be associated with this form of leukemia and or aplastic anemia. In fact, in this country, chloramphenicol usage is very closely monitored. You cannot use chloramphenicol unless you're in very dire straits. We don't use it regularly any longer because of this known association. You do, however, see patients coming in from other countries every now and then, especially from Asia or Eastern Europe, where they're still using chloramphenicol, and you'll see patients come in with this damage. My parents had an exchange student from Albania who, while he was here, they identified he had aplastic anemia from using chloramphenicol when he was back home in Albania. Now, luckily, once we start treatment, most of those patients will survive, but it's a dangerous thing to use chloramphenicol. It's usually only used for things like brain abscesses and those sorts of problems where you really have come to the end of the line and you need to pull out every last stop. All right, quinolones. Quinolones are also very effective antibiotics. Unfortunately, they have been associated with seizures and brain damage, so their use has to be limited as well. Vancomycin has been known to cause liver and kidney damage, just like quinine has. Interesting one with tetracycline. Tetracycline has been shown to permanently stain teeth. In this patient, you can see this. You can see the gray line right here at the base of the teeth. That is permanent. There's nothing we can do to get rid of it. And that comes from using tetracycline when the patient was young. Tetracycline is no longer given to children before the age of about 8 years old because it's known to permanently stain teeth, and there's nothing we can do to undo it, unfortunately. So we don't use it in this country for young kids for this reason. Another possible human side effect to drugs are allergies. Of course, some drugs do cause allergies in certain humans. Some humans are more allergic than others, and they may have allergies against drugs. And allergies can range from relatively mild rashes to deadly allergic responses. This patient is suffering from hives, an allergic reaction. Now, as annoying and itchy and painful as this may be, it's not really life-threatening. What we worry about is allergic responses that start to block airways. That's where we're really concerned. So hives, although they're annoying, are not necessarily very dangerous. But they can get deadly. You can have deadly anaphylactic responses to drugs if you're allergic to them. The most common drugs to trigger allergies are the penicillins and the sulfonamides, the sulfa drugs. Those are the two that humans are most commonly allergic to. Another possible side effect for drugs here. The other possible side effect here is damage to the patient's microflora. Microflora are the normal, natural, healthy bacteria and other microbes that are in and on our body at all times. Let me say it again. The flora are the normal, healthy microbes that live on and in our body. Now, there are microbes that are supposed to be in your body and on the surface of your body, and in fact, without the flora, your body does not function correctly. There are lots of benefits to these floral organisms. Some of the organisms help in digestion. Some of the bacteria and yeast that are in our mouth actually start the digestive process when you put food into your mouth. The bacteria that are in your gastrointestinal tract help complete digestion. In fact, if you don't have the flora in your gastrointestinal tract, you cannot completely digest your food. They're absolutely essential for your digestion. So one of the benefits for flora is digestion. Another benefit, several of the floral organisms that live in our gastrointestinal tract produce vitamins. In fact, they produce vitamins that you don't get any other way. They're not vitamins that you get by ingesting. They're not vitamins that you get because you produce them yourself. They're vitamins that are made by the organism living in your gastrointestinal tract. For example, vitamin K. You get from the E. coli that lives in your gut, produces this vitamin, and excretes it out into the environment, and your body can absorb it. So first benefit to flora is helping in digestion. Second benefit is the production of vitamins. Another benefit here is that flora prevent infection by pathogens. And they do this really two ways. One way they do it is by simply being there. They sort of compete with the pathogen. Because your gastrointestinal tract has a lot of these normal, natural, healthy floral organisms in it, it's hard for a pathogen to set up infection. There's already a bunch of bacteria there that are doing benefits. So one way that they protect you against infection is by simply competing with the pathogen. But new evidence suggests that some floral organisms actually aggressively go after pathogens. For example, they've recognized now that E. coli, who lives in our gastrointestinal tract, when salmonella shows up, E. coli specifically makes chemicals to kill it. So, fascinating. In some ways, they're just simply competing with the pathogens, but in other ways, they aggressively go after the pathogen for you. So, another benefit to flora is that they protect you against infection. So the flora, again, good for you. You gotta have them, otherwise you're not going to digest properly. You're not going to get vitamin production. You are left open to possible infection without your floral organisms. Well, unfortunately, those antibiotics that are extremely potent, potent means very active in small doses, those antibiotics that are extremely potent and broad spectrum, in other words, they kill everything, they kill gram-positives and gram-negatives, they can also hurt your flora. The drugs we've associated with having this problem are tetracycline, chloramphenicol, and quinolones. Now, really, any antibiotic can do this, but these are the ones that we know are particularly bad at killing your flora. So if you treat a patient with something like chloramphenicol, which is a very aggressive killer in very small doses, and it kills everybody. He's going to kill not only the pathogen, but he could also damage your patient's flora. And when you do that, now the patient's not going to digest things properly, and the patient's going to be vitamin deficient, and possibly you're leaving the patient open to what we call superinfections. And a superinfection is brought on by damage to the flora. Let's look at how that works. All right, so here we have, here's our patient. Our patient is suffering from a throat infection, a bacterial throat infection. And if we were to look at the patient's gastrointestinal tract right now, we would find that they have lots of normal, healthy floral organisms. And in this diagram, they're showing you flora in these pink triangles. So lots of healthy pink flora in the gastrointestinal tract. We expect them to be there. Now, at any one time, you're going to have lots of healthy flora, but you may also have one or two pathogens that are sort of passing through. You ate something, it had a little salmonella cell on it, it went through your gastrointestinal tract, so there's always going to be an occasional pathogen passing through. Why isn't that pathogen starting an infection? They're showing you the pathogens in blue here. Why isn't that pathogen starting an infection? Because there's so many healthy pink flora here that the blue pathogen can never get a foothold to start the infection. So the flora is protecting this patient. Now, these pathogens, there's no telling what it is. They may or may not be antibiotic resistant. Some could be resistant to antibiotics, some may not. There's no telling what they are. So again, if we have our patient here, our patient has a throat infection, the patient's flora is here, it's normal, it's healthy, there may be a pathogen or two passing through, maybe one of those pathogens happens to be antibiotic resistant. Now, because of the throat infection, you decide to treat your patient with an antibiotic. You treat them with an antibiotic, and the antibiotic does a great job of killing off this infecting pathogen. Unfortunately, the antibiotic does a great job of also killing off any bacteria in the body that is sensitive to that antibiotic. So it kills off all of the flora. It would also kill off any sensitive pathogens. The only organism that's going to survive is a resistant pathogen. Now, is the resistant pathogen going to have any competition from the flora? No. So now that resistant pathogen is going to grow and multiply and cause a brand new infection that we call a superinfection. So the superinfection actually comes from treatment with an antibiotic that kills off the flora. Now, if we wanted to treat this patient with an antibiotic to kill off his super infection, is it going to work? Not the same one that we used here, because this one's now resistant, and we know it's resistant because of how this developed. So super infections are infections that arise from destruction of the flora. The flora are there to protect us, and if you treat your patient with an antibiotic that kills off the flora, now they can wind up with one of these super infections. All right, another possible damage that can occur from treating with antibiotics. So, we tell patients, don't want to take antibiotics unless absolutely need to, because you don't want to leave yourself open to these possible complications. So all of this information together, how do we choose a drug? How do we put this together and decide what drug we want to use to treat a patient? First thing that you're going to think about, of course, is the nature of the pathogen. What are you fighting? Is it a gram-positive infection? Is it a gram-negative infection? Is it a fungal infection? Is it a parasitic infection? You got to know what you're trying to kill. Second thing you want to think about is the susceptibility of the pathogen. Do you already have reason to believe that this pathogen is resistant to antibiotics? If you do think so, then you may want to use a more aggressive drug. If you have no reason to suspect that it's resistant, then you can use a less aggressive drug so that you don't possibly hurt the patient. So when we choose a drug, we think about the nature of the pathogen. We think about how susceptible that pathogen is, if we have any information about that already. And the next thing we're going to think about is possible toxicity to the patient. Of course, you want to know, is your patient allergic to drugs? You will never choose a drug that is known to have serious side effects unless you have to. You always save that one for worst case scenario positions. How else do we go about choosing a drug? You can use a test called the Kirby-Bauer, or the minimum inhibitory concentration, to test your pathogen susceptibility to various antibiotics. Now, we did the Kirby-Bauer in lab. Remember, in lab, you took that Mueller-Hinton agar, you swabbed the surface of the Mueller-Hinton agar plate, and you put the little antibiotic discs on top of it. Next week, we're going to see the results from that Kirby-Bauer, and we'll look to see how effective each antibiotic is at killing our pathogen, whatever you swabbed on the plate. We can also use another technique to test that called the minimum inhibitory concentration. I'm going to show you how that works in just a moment. And that will test how susceptible our pathogen is to our antibiotics. And then once you have that information, the other thing we want to calculate is called the therapeutic index. The therapeutic index compares the toxicity of a drug against the pathogen susceptibility to the drug. I'm going to talk more about that in just a minute. Let's look at the minimum inhibitory concentration as a way to test how sensitive or resistant our pathogen is to a particular drug. Now, currently, medical professionals are using the minimum inhibitory concentration more than they use Kirby-Bauer, and that's because the MIC is more sensitive and quantitative. Kirby-Bauer sort of tells us, yes, this is an effective way to kill this bacteria, or no, this is not an effective way to kill this bacteria, while the MIC will actually give us a number. It will say how much of the drug do we have to use to kill that pathogen. So it's more sensitive a test. It's also more quantitative. We'll get quantitative data from the MIC. It is more expensive. It requires more media, but you do get better data from the MIC. So here's how it works. To run a MIC test here, you're going to have a whole series of test tubes. Each of them has a varying amount of your antibiotic. Let's say we're going to test ampicillin. So you have a whole bunch of different tubes here. Each tube has a different dose, a different concentration of ampicillin. This one's our control. It has no ampicillin. This one has 0.12 micrograms per mil, 0.25 micrograms per mil, half a microgram per mil, 1 microgram per mil, 2, 4, etc. So you have varying amounts or concentrations of your antibiotic. Then you put the pathogen, the same amount, a loop full of pathogen, into each tube. And basically, you look for the smallest dose that kills the pathogen. So, what's the lowest dose here that kills the pathogen? 4. In this example, 4 micrograms per mil is the smallest dose that kills the pathogen. Because if you only give 2 micrograms per mil, then you get turbidity, you get growth. But if you put 4 micrograms per mil, you have no growth. So the minimum inhibitory concentration, or the MIC, is 4 micrograms per mil. Right? That's the minimum amount you must give to kill off that particular pathogen. So the minimum inhibitory concentration is the smallest concentration of the drug that inhibits growth. It's a quantitative number. It tells us how effective the drug is at killing that particular pathogen. Now, once you have the minimum inhibitory concentration, you can use that data to calculate the therapeutic index. Now, the therapeutic index compares the drug's toxicity to humans to its effectiveness against the pathogen. So, I'll show you an example in just a moment. The therapeutic index is the toxic dose, how much is damaging to the human, divided by the minimum inhibitory concentration. So for example, if we look at drug 1 here, the toxic dose for drug 1 is 10 micrograms per milliliter. In other words, if you give your patient this concentration, 10 micrograms per mil, it's going to be toxic to the human. You're going to have liver problems or kidney problems or whatever it is, brain seizures. You're going to have some toxic issue in your patient. So this is the toxic dose, how much is known to cause problems in humans, known to be toxic to a human body. And then here's the minimum inhibitory concentration. The minimum inhibitory concentration, again, is how much you must give to kill that pathogen. Right? We saw that a moment ago. So the therapeutic index is the toxic dose divided by the MIC. So 10 divided by 1 means the therapeutic index for this drug is 10. If we compare that to another drug, here's drug 2. And drug 2, the toxic dose is the same, 10 micrograms per mil. If you give 10 micrograms per mil, you're going to cause harm in your patient. But for drug 2, the minimum inhibitory concentration is 9 micrograms per mil. In other words, you have to give 9 in order to kill the pathogen. But if you give 10, you damage the human. So 10 divided by 9 is a therapeutic index of 1.1. This is a very dangerous drug. This is a very risky drug. You don't want to use this drug. The therapeutic index is just too low. In other words, you have to give 9 to kill the pathogen, but if you give 10, you're hurting the human. That's a dangerous situation. So a higher therapeutic index is always safer for the patient. We always want a higher therapeutic index. Bigger difference between the toxic dose and the minimum inhibitory concentration, so that you can give plenty of drug to kill off the pathogen without possibly harming the human, without causing toxic problems to the human. Now, the last topic I want to talk about in this area is drug resistance. Of course, drug resistance is becoming a huge problem in medicine and in microbiology. We're seeing lots of problems with bacteria becoming resistant to various different kinds of drugs. And really, if you think about it, how does a bacteria become resistant to a drug? It all starts with random mutations. This is evolution. This is natural selection. Now, that bacteria, just through random mutation, has created a way to survive better in its environment. Now, the first bacteria that ever became resistant to that drug did so through a random mutation. But remember, bacteria can pass DNA from one another. So once one of them has evolved through random mutation, this drug resistance, that bacteria can pass it to others in the environment. So once one of them has evolved this resistance, he can pass it on to others in the environment. How does an adult cell pass DNA on to another adult cell? Transduction, transformation, conjugation. So once one bacteria has become resistant through random mutation, he can pass that gene on to others through transduction, transformation, conjugation. This is a small map that's showing documented interactions. So each arrow here is showing how we know DNA has moved from one organism to another, and this is one small portion of a very large chart showing all documented interactions. So for example, we know that Klebsiella has given DNA to E. coli. We know that E. coli has given DNA to Klebsiella. We know that Haemophilus has given DNA to E. coli. We know that E. coli has given DNA to Shigella and Shigella to E. coli. And we know E. coli has given it to Pseudomonas, and we know that Pseudomonas has given it to Neisseria. We know that Pseudomonas has also given it to Vibrio. We know that Staph has given it to Bacillus, and we know that E. coli has given it to Bacillus. Now that's very unusual because they're not closely related. We know that Serratia has given it to E. coli, and we know that E. coli has to Bacteroides, and Bacteroides back to E. coli. And on and on and on the list goes. This is just one small portion of a large chart showing all of the documented interactions. So if E. coli randomly creates a resistance gene through random mutations, he can very quickly pass it on to Shigella and to Salmonella to Pseudomonas and Pseudomonas to Neisseria, and on the list goes, and they can share this information. And before you know it, they all become resistant to the drug. So what does it mean? When we talk about being resistant to a drug, what does that really mean on a microbial scale? Like, what is it that the cell can do to make it resistant? There's 5 different ways that we know bacteria can become resistant. First one is by inactivating the drug. We talked about this with penicillin. Remember penicillin here? Some bacteria have evolved this enzyme called penicillinase that breaks down the penicillin. If the bacteria has evolved an enzyme to inactivate a drug, that's one way to become resistant. You're just inactivating the drug when it shows up. Another way for bacteria to become resistant, sometimes they block entry of a drug. Drugs come into the cell through little receptors, little tunnels. Well, if the bacteria has mutated its receptors, now the drug never comes into the cell. Right? Then he's resistant. Some bacteria create drug pumps, where the drug comes in, but they have this extra pump that immediately pumps it out. So yes, the drug's coming in, but they pump it out immediately. It never kills the cell. Drug pumps apparently are very, very successful. There are multiple drug pumps. There are some bacteria that have these drug pumps that can pump out 5 or 6 or 7 different classes of antibiotics, which is scary because they become resistant to multiple drugs very, very quickly when they get these drug pumps. Another thing that bacteria can do to become resistant, they can alter the binding site of the drug. If, for example, the drug binds to one portion of the ribosome, in this case, one portion of the large subunit of the ribosome, well, they just mutate their ribosome a little teeny bit, and the drug no longer binds. So they alter the binding site of the drug to become resistant. Another possibility is to use an alternate pathway. If a drug is targeting one particular pathway of the bacteria can evolve a new way around it. So these are the ways that we know bacteria can become resistant. They can either inactivate the drug, block the drug from entering, pump the drug out once it's entered, alter wherever the drug binds to, or just completely make a new pathway, evolve a new way around. So when we talk about a drug resistance gene, it's doing one of these things. The gene is either making a protein to break down the drug, or they're changing the receptor, or pumping the drug out, or changing the binding site, one of these different possibilities. Now, of course, once the bacteria have become resistant, originally through random mutation, when we use antibiotics, we really only encourage them to develop that resistance. This is natural selection. If we put pressure on the bacteria to evolve the resistance, then resistance becomes more common. For example, Here we have a whole population of microbial cells. In black are the normal microbial cells, not resistant, and maybe one or two of them have evolved resistance, right? They happen to have a random mutation where they have resistance. Now, as long as there's no drug around, there's no benefit for these that happen to have resistance. So they may be there in low numbers, but not really a problem. However, if we expose this entire plate to an antibiotic, the only ones that are going to survive are the resistant ones. Therefore, the next generation is more likely to be resistant. In other words, the more we expose bacteria to the drugs, the more likely they become resistant. And that's the horrible reality of bacterial resistance to antibiotics, is the more we use antibiotics, the more we try to kill them off, the more we encourage them to become resistant. And that's the really horrifying fact of the matter, is that we're forcing them to evolve. We are encouraging them, at the very least, to evolve to become more resistant through our use of antibiotics. So the cause and effect here of resistance. Resistance is an issue in all environments. Of course, in the body is first. If you use an antibiotic to kill off bacteria, you're only killing off sensitive bacteria. That means you're leaving resistant bacteria, which means the more we use drugs, the more likely they are more likely bacteria are to evolve resistance against those drugs. And that's a serious problem in human bodies. In a hospital environment. A hospital environment is really the perfect place to evolve resistance. If you think about a hospital environment, in the hospital we have all sorts of different pathogens because that's where we put our sick people who are infected. So we'll have lots of different kinds of pathogens there in the hospital. We also have lots of different kinds of drugs. So the pathogens are constantly being exposed to multiple different kinds of antibiotics, which means we are encouraging those pathogens to become resistant by constant exposure to antibiotics. And of course, the hospital is also full of the sickest of the sick people. So we put the sickest of the sick people in an environment that has lots of antibiotics and lots of pathogens, and they are more likely to get an antibiotic-resistant infection in a hospital than anywhere else. In fact, if you want to get an antibiotic-resistant infection, your best bet is to go to the hospital. That's where you're most likely to get it. Unfortunately, in the livestock, we're also seeing significant resistances because many ranchers are now putting antibiotics in the livestock feed, the feed that they give them, and what they're trying to do is increase the health of their stock. But unfortunately, what happens is the antibiotic will not only encourage resistance in the animal, but also, as the antibiotic goes through the animal, it comes out in the feces, it comes out in the urine. Where does the urine and the feces go? Into the soil. And so we encourage antibiotic resistance in the soil. And we're now seeing lots of soil bacteria that have multiple drug resistances as well, because they're constantly getting exposed to antibiotics through livestock, feces, and urine. Additionally, the soil eventually washes off into the water. And now water samples have antibiotic-resistant bacteria in them as well. And now that we're looking for them, you find them everywhere in the soil samples and water samples. Really, we have antibiotics surrounding us constantly, and therefore the bacteria are evolving resistance. That's just natural selection. That's evolution. Other things that encourage antibiotic resistance. Inappropriate antibiotic prescriptions. We in this country, really all over the world, but if we just think about this country for a moment, in this country we overprescribe antibiotics at an alarming rate. About 75% of all prescriptions that will be written this year, 75% of those prescriptions will be for an upper respiratory tract infection, a cold. Well, almost all upper respiratory tract infections are viral infections. So is that antibiotic, which is meant to kill bacteria, gonna do anything for your viral cold? Of course not. The answer is no. Okay? So we have no business giving prescriptions for antibiotics to kill bacteria if the patient likely has a viral infection. Not only are you not really helping your patient at all, you're also increasing the likelihood of evolving more resistance if you keep exposing bacteria to those antibiotics. So this is gross over prescription of antibiotics. And now the question, of course, is why? Why do we prescribe antibiotics when they're not particularly useful? And there's a couple of reasons for that. First off, it is very difficult when you have a parent in front of you with a screaming baby and the baby has a cold and you're trying to explain to the parent that the baby has a viral infection and the antibiotic's not going to help them, and they don't want to listen. They want drugs. They want an antibiotic. Give me the antibiotic now. And, you know, it's easy for me to say they shouldn't do that, but when you're actually in the position of being faced with somebody who's saying this to you, it's much easier just give them the prescription and move on with your day. And unfortunately, that happens way too often. Again, it's easy for me to say I don't have to be face to face with this scenario on a daily basis, but that unfortunately is a very difficult thing because it's increasing the resistance against antibiotics. And now the other thing I do want to point out, sometimes it is appropriate to give an antibiotic prescription prophylactically, not usually. Some doctors will give antibiotic prescriptions prophylactically, like we think you might have an infection, so I'm going to give you this antibiotic. I'll call you in a couple of days and let you know if you should keep taking it, those sorts of things. That's generally a very bad idea. You don't want to give patients antibiotics unless you know they need them because you don't want to encourage resistances. There are exceptions to the rules, however. There are patients that we know are prone to secondary infections. They get one infection and then bam, they get another one on top of it. If the patient has a history of that, then even if they have a viral infection, it would be worth giving them an antibiotic because you know their history of getting multiple infections at the same time. So there are some times when it's appropriate to give prophylactic antibiotic prescriptions, but generally you shouldn't do that. Generally you want to only give it when you know it's bacterial and when you know that you can cure it through that antibiotic. Another problem here, inappropriate antibiotic usage. On inappropriate antibiotic usage, I'm really talking about the patient's perspective here. It is so important for patients to completely finish their antibiotic prescription. No stopping treatment early. Right? You must use the antibiotic exactly as it was prescribed, and you must finish the entire prescription. And the reason for that is many times as bacteria evolve resistance, they're sort of slightly resistant at the beginning. And if you keep treating with the antibiotic, you will kill them off in that 10 days or 2 weeks of antibiotic treatment. But if you stop early, you encourage them to keep evolving the resistance. So you never do that. If you start an antibiotic treatment, you must finish it. Even if you feel better in a couple of days, you must finish that antibiotic because you don't want to encourage the evolution of resistances. Also, there's none of this sharing leftover antibiotics. I mean, you shouldn't have leftover. You should finish your treatment, but there's none of this, oh, well, you think you have a cold? Take some of my antibiotics that I have in my medicine chest that have been sitting there for a year and a half. No. You don't do that. You don't use other people's prescriptions, especially older prescriptions where they're not as effective and therefore they encourage resistances. You want to finish the entire course. You won't have enough to finish an entire course. You need to go in and get your own antibiotic prescription for your particular infection. If you, for whatever reason, do have leftover pills, which you shouldn't have because you should be finishing your prescription, but if there are leftover pills, you never flush them down the toilet. That gets antibiotics into our soil samples. You can throw them away in sealed containers. That's the best way to get rid of them, or contact local hazardous waste disposal. I know it sounds excessive, but don't ever flush antibiotics down the drain. We already get enough of it in the water samples because of urine, so you don't want to put any more down the drain. So again, inappropriate prescriptions on the doctor's side and inappropriate usage on the patient side is causing a bigger problem with resistance as well. And of course, the other thing on the antibiotic prescription side is doctors should never prescribe a newer drug if an older drug will work. As we create new drugs, we don't want to encourage resistance to the new drug. So it's always a good idea to keep using the old drug until it doesn't work anymore, and then switch out to the new drug. You don't want to switch until you have to. You want to make sure that you are keeping some drugs on the shelf for worst case scenarios where nothing else is working. So how bad is the problem? I know this data is actually kind of old in comparison, but it's such a striking difference in a couple of years time frame that I like to show this chart. This is the rise and the increase in resistant bacteria within just a couple of years, really. So for example, if we look at pneumococci, this is Streptococcus pneumoniae that can cause pneumonia and ear infections and in some cases meningitis. In 1989, you almost never saw penicillin-resistant strep pneumoniae. By the year 2000, 30% of patient isolates were resistant to penicillin. That is really fast. In 11 years, 30% became resistant. By 2002, the number was at 40%. That's a striking increase very, very quickly. If we look at something like Neisseria gonorrhea that causes gonorrhea, here, gonococci down here, penicillin, about 10% of patients were resistant in 1990, 70% by 2000, 74% by 2002. That is a scary increase very, very quickly. And again, the reason for this is the more we expose the bacteria to antibiotics, the more they're going to evolve resistance. That's natural selection at work. So what can we do? The future of antibiotics here. The future of antibiotics, the first thing is combined drug therapy. This is a new approach to try and deal with antibiotic resistance. The idea behind combined drug therapy is use several antibiotics at the same time, and hopefully the bacteria can't evolve resistance to multiple drugs all at the same time. In other words, throw lots of drugs at it all at once. Something's going to kill it. There's no way he can evolve resistance to everything all at the same time. That does work at killing the bacteria, but as you can imagine, it wreaks havoc on a patient's flora to have multiple drugs given at the same time, and it can make you feel really, really terrible. So combined drug therapy will work on very severe cases of resistance, but it's not a pleasant therapy for sure. Continual search for new antibiotics. Of course, we are continually looking for new antibiotics. Most new antibiotics come from soil samples, identifying soil bacteria or fungus that are producing something to kill off other soil microbes. So they are constantly searching for new antibiotics. Of course, eventually we're going to find them all, and the well will run dry. The other problem is that it really doesn't give much money to the company. Unfortunately, the research and development of new antibiotics is slowing down. And if you think about from a drug company's perspective, there's really just not much money out there in antibiotic treatments. A drug company, just like any other company, they have employees they have to pay, and they have stockholders who want a profit. And so they are not an altruistic governmental agency, they are a company. And from their perspective, if they spend millions of dollars in research and development to develop a brand new antibiotic, and then 3 or 4 years after the antibiotic's out there, it's no longer effective because all of the bacteria are resistant to their antibiotic. It just doesn't make good sense for their bottom line. It just doesn't make any good money sense, because as they spend all this money developing it, the bacteria will evolve resistance and their drug will no longer be useful. So quite frankly, most drug companies have stopped looking for antibiotics. They are not they're not money makers for the drug company. In fact, they're money losers, and so they don't want to spend their time and effort there. They'd much rather look for drugs that will do something like cure cancer, that will be useful long term, or Viagra, right, to cure erectile dysfunction, where you're not going to evolve resistance. Headache medications, these sorts of things where you don't have the same resistance problem that you do with antibiotics. So, luckily the government is funding some research on looking for new antibiotics, so the research is ongoing. The government funds it, luckily, in this country. Not very many other countries fund that work, but the United States government does. National Institutes of Health funds the search for new antibiotics, but again, it is slowing down because companies are no longer doing it for understandable reasons. And eventually we will find them all. So when it comes down to it, the future of antibiotics is not a good one. We know that this is sort of a sinking ship that we're on. We are trying to kill off these organisms using a drug, and they're going to try to evolve resistance, and we will never win this war. We're never going to win the war and kill them off entirely, and they will continue to evolve resistance. So the question is, what are we going to do? And the answer is we don't know yet. Unfortunately, I'm leaving you with a very scary scenario, and I'm sorry if you have nightmares. But the truth of the matter is we don't know what's going to happen. We know that what we're doing now is not going to work long term. And we know that, in fact, not just long term, but within the next couple of decades, we're really going to have to change how we do business, because antibiotics are a sinking ship and they will not work forever. In fact, they may not work for another 100 years. So we really have to come up with new treatment plans. What can we do? There are some ideas out there. Some researchers are thinking about creating viruses that attach to receptors only on pathogens, so that you can treat your patient with this bacterial virus, which would destroy just the bacteria you want it to destroy. You can imagine how difficult it is to convince a patient that I'm going to infect you with a virus to help your infection. They're not really keen on that idea. Another possible idea out there right now are probiotics, where we try to provide some more floral organisms to help compete with the pathogen. And these are some of the current ideas on how to treat patients. And when it comes down to it, we're still figuring it out. It will be in your careers as healthcare professionals that we have to tackle this problem, and we will have to tackle it because the ship is going down, and we're going to have to come up with a new way to treat bacterial infections, because antibiotics are not going to last long. All right, well, that's the very sad downer that I'm leaving you with for this lecture. Don't forget, for our next lab time, you need to prepare for the peer teaching activity. And I will see you guys next time.